Hi everyone, this is Vamshi Krishna, AGPSC third year from Agricultural College, Jagitya. Today we are going to discuss about backcross. Backcross method of breeding was developed by the Harlan and Pope. It is to take out the undesirable allele at the particular locus is replaced by the desirable allele. If there is one or two undesirable alleles in case of a good variety, so that undesirable alleles must be replaced by the desirable alleles. For that purpose, it is developed. As in the olden days, in the early 20th centuries, they used the pure line selections for the disease resistance. As the interspecific and intraspecific pure line selections are inefficient for the disease resistance, so that the backcross method is developed to make this desirable with good resistance against the diseases and pests. Further, let us move into the backcross method. First, let us know about the backcross. What is meant by backcross? Backcross means crossing of F1 hybrid with any one of its parents. So let us see here. Let us take two parents, capital T, small t. This is the recessive parent and it is the dominant parent. So we get the heterozygous. It is a tall and this is dwarf, and we get heterozygous tall. So when this F1 hybrid crosses with this recessive parent or this dominant parent, it is called as a backcross. The backcross with the recessive parent, it is also called as the test cross. So in this way, all the test cross are backcross, but all the backcrosses are not the test cross. So let us move what is meant by backcross method. In this backcross method, F1 it is F1 and the subsequent generations. All the subsequent generations are also backcrossed with the parents. It is known as a backcross method. What is the need of backcross method? It is the main objective of the backcross method is to improve or correct the some of the specific defects which are present in the a good high yielding variety with all other desirable characters. So now let us know about what are the requirements for the backcross method. The backcross method requires a good donor parent with one or two specific characters and a recipient parent which is high yielding and good with the all other agronomic traits except for one or two specific characters, and the character or the gene which is going to be donated, it must be highly heritable and high expressivity. It must have the high expressivity. These are the requirements. And now the recurrent parent and non-recurrent parent, let us know about. The recipient parent is also called as a recurrent parent, as it is repeatedly used in case of the backcross method for getting the characters from the donor parents into the recurrent parent. And non-recurrent parent is the donor parent which is used only once in case of this backcross method. And the applications of the backcross, let us know about the applications of backcross. It can be used in case of intervarietal transfer of simply inherited characters or quantitative characters. In case of simply inherited characters like disease resistance, and in case of quantitative characters, intervarietal. Let us know about what is meant by intervarietal. It is a cross between two varieties. Example, JGL 24423 crossed with something RNR 1508 variety. It is the intervarietal. These varieties are belongs to same species. And quantitative characters are earliness and panicle initiation, height. Next one, interspecific. Interspecific means species are different. Let us know about some of the Gossypium hirsutum and Gossypium arboreum. It is a interspecific, or Vigna radiata and Vigna munga. This is the green gram and black gram. Different species and the genus is same. Gossypium hirsutum, Gossypium arboreum. These species are different but genus is same. Interspecific crosses. for the inheritance of simply inherited characters. Earlier only I have said that backrows is for simply inherited and quick high expressivity characters are used in case of backrows. For that example, for interspecific transfer of simply inherited character, Blackham resistance in case of Gospeum species is transferred to the Gospeum hirsutum. Several Gospeum species have the Blackham resistance and it is transferred to the Gospeum hirsutum. And in case of wheat, leaf and stem rust is transferred from Triticum monococcum to the Triticum astrum through backcross method, and for development of CMS and CGMS lines through the transfer of cytoplasm by the backcross method, and for getting of transgressive segregations. This transgressive segregations is in case of backcross we get more heterozygosity in F1 and F2 progeny, and in this we can go for the selections in the segregant populations. And next one, for the production of isogenic lines for gene studies, how one gene is controlling isogenic lines means two lines with except one character is different, those two lines are called as isogenic lines. And for that gene we can go for the study of the gene studies. And next one, germplasm collection. For germplasm collection, if we go for photosensitive or thermosensitive germplasms, then we will restore them with the backcross method. And in case of backcross, we can go for single gene transfer or multiple gene transfers. And based on the type of transfer, there are three types. Simultaneous transfer. Simultaneous transfer means more genes, one or two genes are transferred at a time. The advantage of this method is if there are the desirable linkages. If there is a desirable linkage between the two characters, the gene can be transferred easily. Example for the desirable linkage is LR24 and SR24. And if there are undesirable linkage between the two genes, then stepwise transfer is preferred, as one gene can be transferred once and then another gene. Stepwise transfer means one gene first enter can be sent into the recurrent parent, and then again another gene is sent into the recurrent parent by the backcross method. Another, it is next one, it is first one. And then simultaneous but stepwise transfer, one character is transferred into one of the recurrent parent, and then second gene is transferred into recurrent parent, and these two recurrent parents are crossed to get both the characters. First character and second characters can be inherited simultaneously but stepwise. And let us know about the what are the advantages and disadvantages after the discussing about this transfer of dominant gene and transfer of recessive gene. Now let us know in detail about the transfer of dominant gene. Now here the dominant gene is controlling or giving the resistance to the one of the strain. The strain B is resistant to stem rust in case of wheat, which is dominant gene, and the susceptible is stem rust susceptible is with good high yielding variety. It is strain A, and the strain A and strain B are crossed in the first year. The strain A is the recurrent parent as it is lacking the resistance, and the strain B is having the resistance and it is donating the gene to the recurrent parent, so it is a donor or non-recurrent parent. And this recurrent parent is recipient parent as it is receiving the gene from the strain B. And in the first year, the non-recurrent parent and recurrent parent are crossed, capital R crossed with the smaller, so we get the progeny heterozygous, capital R and smaller. So all this progeny contains the disease resistance, and uh, this F1 hybrid is again crossed with the recurrent parent, so it is a backcross one. Recurrent parent smaller, we get the progeny is like smaller, capital R smaller and smaller. Let us see here. Here capital R smaller, capital R smaller, smaller smaller, capital R smaller, and smaller smaller. We get 50% of disease resistance and 50% of susceptible progeny. So we must discard the susceptible progeny. Half of the resistant progenies and half of the susceptible progeny. So half of the resistant progeny are selected and half of the susceptible progeny are discarded. And then again it will be going for the crossing with the recessive parent or recurrent parent. And then it leads to the progeny. Same the progeny is here, and the 50% of the susceptible parent are discarded and the resistant 50% progeny are selected, and it is repeated up to B B6 backcross 6 up to backcross 6. Here up to backcross 5 we are going for the selection, and up to backrow 6. In the backrow 6 around 98.4% of the genes are transferred. In the first year around 75% transfer, and then later and 88 percentage, and up to 60 up to backrow 6 we get around 98.4% of genes of resistant genes are transferred into the strain A or variety A, and the resistance is developed in the variety A. So the resistant rust resistant progeny are selected and harvested separately and grown as individual plant progeny rows, and in this individual plant progeny rows which are homozygous for the rust resistance are harvested separately, and this constitute as a new variety, and this new variety is grown again as individual plant progenies, and in this the select and again this progeny is selected and sent for the replicated yield trials with the check A. Check A is susceptible, and all the progenies which are selected in the backrow 6 and F3 generation and backrow 6 F2 generation are checked with the variety A in the replicated yield trials, and then it is further went for the multiplication and then distribution to the farmers in the 12th year. And next one, let us going to discuss about the transfer of recessive gene. Here the resistance is due to the recessive gene. So after every backcross, in case of backcross 1, after the backcross 1 and for every 2 backcrosses we go for F2 progeny, and in this F2 progeny we are going to identify the rust resistant progeny, and we can go for cross again we go for the backcrosses. So let us see how it will be transferred. The recessive genes are transferred. The non-recurrent parent uh, is recessive genes smaller and recurrent parent which is the dominant genes capital R and capital R. These two are crossed and we get the F1 progeny capital R smaller. This F1 hybrid is susceptible. All the F1 are, when we cross it with smaller capital R we get the capital R and smaller. In case of this recessive gene is resistant, so this F1 progeny is susceptible to rust. So the no rust resistant test is conducted in the F1, and it is crossed with the recurrent parent in the backcross one recurrent parent. So we get the when this is crossed with the recurrent parent, we get the progeny of capital R and capital R smaller. Both are rust susceptible varieties. So in this backcross one progeny we can go for backcross F2, and this in backcross 1 the non-rust resistant there is no rust resistant, and these are self due to the self pollination. In case of heterozygous progeny we get selfing, we get the capital smaller capital smaller. In these three populations these two are not rust resistant, so these two are discarded and these rust resistants are selected. Here you can see when this is selfed we get only capital R, and when this heterozygous is selfed we get the capital R smaller and smaller. These two dominant genes are rust susceptible. So these two are discarded, and this smaller is rust resistant. So this is selected. Here the rust, with the help of rust test, we inoculate the rust spores into the field, and we select the rust resistant recessive parent, and it is back crossed with the dominant capital R strain A. And again we get the heterozygous capital smaller, and this is crossed again with the recurrent parent capital R. Here there is no need of rust resistant test, as it is susceptible to the rust, and then it is again gives the progeny in the backcross 3 as a capital R rust susceptible, and capital R smaller, it is also rust susceptible. So there is no rust resistance test, and this is again go for the F2 progeny. Initially I have said that after every two back crosses we go for the selection. In the back cross one, immediately after back cross one we go for the selection in the F2, and after every two back crosses we can go for the F2 generation and selection. And in this, when the capital is selfed, we get the capital R, and it is dominant and it is rust susceptible, so it is discarded. When the heterozygous progeny is selfed, we get the capital and capital smaller and smaller. So this heterozygous is also rust susceptible, so it is discarded, and the rest two are selected and back crossed with the strain A, and we get the progeny capital smaller and capital R, and this is there is no rust test here, and again the progeny is dominant, so it is susceptible. And again we go for the backcross 5 F2 progeny. Here we can go for the rust epidemic. It is the rigid selection, it is very important in case of dominant, in case of recessive gene. Backcross 5 F2 progeny, here the rust epidemic condition is created, and the rigid selection for the rust resistance is selected, and self-food and harvested separately the seeds and grown as individual plant progeny rows. And here the rust epidemic is there, and in this individual plant progeny rows we go for the rigid selection, and we go for the replicated yield trials, and go for the multiplication and release of the variety with the rust resistance in, in, in the progeny. And now let us know about the merits and demerits of the backcross. The first merit is the new variety is identical to that of recurrent parent except one character. So we need not to go for the testing of the variety. And small population, we require less population compared with the pedigree method for the backcross method. And next one, any defects, one or two defects can be either susceptibility of the disease resistance, susceptibility of the disease can be corrected here. And interspecific gene transfer or cytoplasm transfer can be done. And the demerits are the new variety is not superior over the recurrent parent except for one character, and if there is undesirable gene linkage, the undesirable gene can be transferred into the recurrent parent with a good characters, good agronomic characters. And hybridization, we can hybridization is a big problem in case of backcross. For every backcross we go for hybridization, uh, crossing with the recurrent parent. So every backcross requires the hybridization. It is a time consuming process, and it takes much time. It takes around 12 to 13 years. So in this span we get the new varieties which can replace the recurrent parent. So it is of futile when we get the type of new varieties which are superior over the recurrent parent. Thank you. I hope everyone will like this video and subscribe.